Hello all. So today the topic is related to molecular biology, a very small portion that is genetic material, and also it is the very basic thing we have to know when we get into molecular biology. So what is a genetic material? It's a molecule, and what is its function? It stores and transmits hereditary information, and also it is from one generation to the next. So it is a molecule that stores and transmits hereditary information from one generation to the next. And it's also the base for the growth, development, metabolism, and reproduction of an organism. So it is the information stored in the genetic material that decides the growth, development, metabolism, and reproduction. And we have all heard about DNA. So it is the most common, or in most living organisms, the genetic material is DNA. By the way, in some viruses, RNA acts as genetic material. Now let's take a look into the history of genetic material, some discoveries related to this. And the first one is during 1869 by Frederick Mischer. He isolated a phosphorus-rich substance called nuclein from WBC. It was later identified as DNA. In 1928, Frederick Griffith discovered bacterial transformation, suggesting the existence of a transforming principle, and that also was later identified as DNA. In 1944, Avery, McLeod, and McCarthy demonstrated that DNA is the transforming principle. And in 1952, Hershey and Chase confirmed that DNA is the genetic material used in bacteriophages. Now let's take a deeper look into the Griffiths experiment 1928, and it was about the bacterial transformation. He was working with the Streptococcus pneumoniae, the bacterium that causes pneumonia. He identified two strains, that is S strain and R strain. S strain, as the name suggests, it is a smooth strain that produces a polysaccharide coat which is pathogenic, that is virulent, and R strain that has no coat, non-pathogenic, and does not cause disease. He conducted 4 experiments, in which the first one was with the smooth strain he injected into the mice, and the mouse died. And the S strain was discovered from the dead mouse. So it was concluded that S strain is virulent or pathogenic. And in the second experiment, he used the rough strain and injected into the mice, and the mouse lived. And the R strain is concluded as non-virulent. And in the third experiment, he used heat-killed S strain and injected into the mouse, and the mouse lived and no bacteria discovered. And it is concluded that heat-killed S strain is non-virulent. And now the fourth experiment, in which he used R strain plus heat-killed S strain and together injected into the mouse, and the mouse died. And the S strain, which was live, was recovered from the dead mouse. So here a change occurred. The R strain, which was live, has now become virulent, that is turned into S strain. So something from the heat-killed S strain has transformed into the live R strain, which turned the live R strain into the S strain and become pathogenic. So here Griffith concluded that some transforming principle from heat-killed S bacteria had entered the R bacteria and transformed them into virulent S bacteria. So here the transformation is where the non-virulent R strain took up the transforming principle RNA from dead S bacteria and turned into S strain that is virulent. This discovery is very significant as it is the first evidence that genetic material can be transformed from one bacterium to another. And also it is the foundation for identifying DNA as genetic material, which was confirmed by Avery, McLeod, and McCarthy in 1944, which is the experiment we are going to discuss next. Now we are going to discuss about the demonstration that DNA is the transforming principle. So Griffith had already shown that a substance from the heat-killed virulent bacteria could transform non-virulent bacteria into virulent form. Avery and his colleagues found out which molecule is the transforming principle. They used virulent S strain, non-virulent R strain, and enzymes that destroy specific molecules like proteins, DNA, and RNA. In the first experiment, they used no enzyme. So, in the heat killed S extract, they added live R bacteria and the mouse died. So, here the transformation has occurred and the mice had S bacteria recovered from its body. In the second experiment, they used protease treatment. The protease treated S extract had proteins destroyed, and then they added live R bacteria and injected into the mice, and the mouse was dead. And virulent S bacteria was recovered from the mice. So here also the transformation had occurred, and now it is evident that proteins are not the transformation principle. In a third experiment, they used RNAs and treated the heat killed S extract with RNAs, and now the RNAs are destroyed, and now live R strain is added and together injected into the mouse, and the mouse died where the virulent S bacteria was recovered. So here also the transformation occurred, and thus the transformation principle is not RNA. Now, in the fourth experiment, they used DNAs with the heat killed S extract, thus, the DNAs are now destroyed. So, the heat killed S extract with DNAs destroyed is mixed with live R bacteria and injected into the mouse. Here, the mouse lived, so no S bacteria is recovered. So here no transformation occurred, and it is evident that DNA is the transforming principle. In the fifth experiment, they used DNase treatment plus DNase inactivated. So here the DNase was inactivated by heating, and the live R strain bacteria was added and together injected into the mouse, and the mouse died. So the transformation has occurred here also. In the sixth experiment, they used DNase treatment and DNA added back. So here the DNA destroyed and purified DNA were added, and also mixed with live R bacteria and injected into mouse, and the mouse died. So here also the transformation restored. So from the experiments conducted, it was concluded that only when DNA was destroyed did transformation fail to occur. When DNA was inactivated or added back, the transformation was restored. Therefore, DNA is the transforming principle and the genetic material. This experiment was really significant that it proved that DNA is the genetic material and not protein or RNA. This paved way for later experiments like Hershey-Chase experiment that confirmed DNA as genetic material in viruses as well. So the next experiment is Hershey and Chase experiment in 1952, which proved that DNA, not protein, is the genetic material. In this experiment, Alfred Hershey and Martha Chase used bacteriophages as experimental material, which had a protein coat called capsid and DNA as genetic material. Hershey and Chase used radioactive isotopes to find out which component enters the bacterial cell and directs the infection. In the first experiment, the phage protein labeled with sulfur 35 was allowed to infect the E. coli cell and blended to remove the phage coats. The radioactivity in the supernatant and no radioactivity in the pellet was discovered. It was concluded that protein did not enter the bacterial cell. In the second experiment, the phage DNA labeled with phosphorus 32 was allowed to infect the E. coli and then blended to remove the phage coats. Here, the non-radioactivity in the supernatant was discovered and radioactivity in pellet was discovered. It was concluded that DNA entered the bacterial cell and is the genetic material. The overall experiment concluded that DNA is the genetic material, not protein. So, DNA controls the activities of the cell and is passed on to the next generation. So, that's all about genetic material, the major discoveries, and the conclusion that DNA is the genetic material. Thank you for watching and do subscribe for related videos. See you all in the next video.